Good evening, sir.
How are you today?
I'm fine, sir.
All right, we're going to start in the next 2-3 minutes. Let's just wait for some other persons to come on board.
Okay, sir.
All right, then. You're welcome. I'm going to share this evening water analysis. By way of introduction, water has different sources. We can get water from different sources. Some water are called groundwater sourced. Some are surface water. Some are wastewater. Some are rainwater. Some are storm water. When we say groundwater, those are waters that we dig from the ground. Let's admit someone. Okay. Yeah, so when we dig borehole, that one is groundwater. It might be deep well or just a little dug like we have in Lagos, you know. Yeah, so groundwater involves borehole and wells. Surface water are all these rivers, oceans, streams, lakes. Those ones are called surface water. Okay, so rainwater, of course, we all know rainwater, and storm water too, we know that. Okay, so the design of our analysis depends on the source of the water. We need to, if somebody wants us to analyze water, we need to know the source of that water. This water you are giving me to analyze, where did you get it from? Is it from a borehole? Is it from the ocean, the Atlantic Ocean, the Caribbean Ocean? Or is it from a sea or from the stream in your village? So I need such information to know how to analyze his borehole. Okay, the first thing I'll do is to establish my standard operating procedure. And every company, every analytical company has their own standard operating procedure. Once somebody invites us to analyze water for them, this is what we ask for. This is what we tell them to do before we come and all of that. This is how we are going to do it. When we sample again that the water I got the first time is not enough and all of that, and you're not going to take me seriously. If somebody came and took sample from you the first time and is going back again to come and take sample again, I wonder what you'll be telling them happened to the first one and all of that. So the container size is important, okay? And then again, we need to say that some regulations also exist that tells us that the kind of chemicals like sodium thiosulfate, and that's a dechlorinating agent. We use it to stop the reaction of chlorine because some people have already added chlorine to their water before we sample it. So we need to add sodium thiosulfate to stop the activity of chlorine. Otherwise, what we took 3-4 days ago is not what we have today because the reaction of chlorine continued since these instructions on the slides. We'll still let you have the slide at the end of the presentation so you can actually look at it, okay? And it's important to put all of those things in place. Now, documentation. Once you go out there and we take samples from the field, we need to write things down. In the world today, people are not trained to document. Some people do a lot of things, but they don't write what they've done down. And whatever you have not written down means that you have not done it. Don't say you're going to write at a later time. You need to write it as you are doing it. So if you have gone to Nwaniba in Uyo to take samples, write the address of the place you've taken it, write the time, write the quantity you have taken, label everything. Documentation must be very explicit. Okay? And then when you are submitting the samples to the lab, you need to also document. All the chain of custody must be established. In fact, there are forms that are in place for one to write what one has done at a particular time or the other. Okay, we also have field form an impression, an opinion of the kind of water based on what we see at that initial time. Do we have any questions at this point? Everyone?
No, sir, no question.
All right, then. So let's continue.
I have questions, sir.
Okay, let's have your question, please.
Sir, please, under the source of water,
Yes.
what's the difference between stormwater and rainwater?
Okay. Rainwater. Of course, you know rainwater, don't you? Rainwater falls directly on your roof or maybe directly into the container. Okay. And now some of the rainwater doesn't actually fall into a container, that it falls on the ground and start rolling down, moving, finding its way to maybe a river or whatever. Okay, so the one that you don't collect into a container as it's coming up, coming from up down into your container. The one that you collect like that is called rainwater. The one you don't collect that keeps flowing around, maybe into gutter, into whatever. That's what we call storm water.
Okay, sir.
All right.
Thank you, sir.
Now, if we also have what we call wastewater. Have you heard of wastewater? Living cells or living things in their water. But having done that, you're not done yet. How about the chemicals and all of that? The microbes can affect us, yes. But even after we have killed them, the chemicals in there can also cause some health burdens to us. Okay? So what are the waterborne pathogens we have in water? We have bacteria, viruses, protozoa, helminths, algae, So all of these things can be found in different quantities or amounts in water, depending on the source of that water. If the water is from a borehole, from their overhead tank, and they have not cleaned it for a long time, there's going to be some growth of algae. All this green stuff will be there. And sincerely, most people that use such water, they don't even clean their borehole in a year. Bacteria, look at the diseases they cause on the left side, and look at the organisms that can cause all those things: Campylobacter, Vibrio cholerae, E. coli, Salmonella, Shigella, and all of that. And look at the sicknesses they can cause. And I want us to note that the amount of money people spend on curing these diseases on my left here, if you pull it together in Nigeria in 1 year, it might be able to pay for all the monies ASU people are looking for, but we allow all these things happen. People feel hospitals all the time. Doctors are busy treating enteritis, anaerobes, whether they are rod-shaped, non-sporing, non-spore-forming stuffs, lactose fermenters, gas-producing, acid-producing. Now, all these biochemical protocols are put on the side here, are what helps us to know one organism from the other. And so microbiologists use parameters like gas producing or acid producing to describe and characterize the pathogens that have been isolated in the water we are analyzing. Then some water also contains streptococci, enterococci, and all of that. So when we do deep and comprehensive microbiological analysis, these are the things we are going to be looking out for. And then we want to count how many of them we have, how many problems we are facing. Then we can have some viruses present in our water too. Water can carry all these viruses and diseases too. And they can give us hepatitis A, hepatitis E, and all whatnot. Then protozoans, we can have amoeba, We can have a Giardia species in the water. Okay, so we need to analyze our water very well before we drink it. And so we have a challenge today. There are so many boreholes all around us. There are many sachet water companies. And I'm not even too certain that NAFDAC is doing what they should do in this, to this company. Sometimes once they pay and settle, have there, for the next 5 years, it's business as usual. And our lives are at risk. And the truth is, it's only God that can help us. Some water can even carry worms. I'm sure if you see that one, you throw the water away. How about the ones you don't see? Okay? So the eggs of worms and all of that can be found in the water we are drinking. All right? So all of this constitute what we call biological parameters in water analysis. Okay? All right, let's continue. We can also have algae and fungi in our water also. And so our design of water analysis must be able to capture, we must spread the design to be able to capture all of these things. And so if somebody gives us water to analyze, we want to tell the person there is fungi in your water, there is algae in your water, there is streptococcus in your water, there is E. coli in your water. And they want to ask us, what can I do? How can I treat it? Then we can refer the person to a very good water treatment system so that the water the person is giving out can give life, not death. So now let us go to microbiological assessment of water. That seems to be the major concern. How do we microbiologically analyze for microbes in water? There are two major methods. One is called most probable number, MPN, and the other one is membrane filtration. Let me start with that membrane filtration. That one, we have some membranes, just like a filter paper, that we filter the water through. And then having filtered sample of that water, we take that membrane and incubate it and see what kind of organisms grow there. And then we can subculture until we are able to say, these are the kind of microbes we have seen. They're very simple procedures, okay? In a laboratory, you can easily follow and understand and see how this is done. Like I said, you filter the sample through a particular membrane filter that retains the microbes on this reaction in this water. Okay, now, if after the first one, you can see some acid and gas in some of the nutrient broth, they are going to do the second one. This second one, you are going to use a selective differential media that can actually tell us indeed what we have there is E. coli. Because it's not only E. coli that produces acid and gas. Some other organisms, if you have any questions before we go to the last lab, which is the chemical analysis. Any questions?
Yes, sir, I have a question.
Okay.
You were saying something about you would have loved we online to come and have a idea of how to analyze that water in the Bioscird lab. Is there no way you can do a video of the analysis so that we can watch and practice it at home? Like the simplest one we can do on our own without traveling down to Akwa Ibom for the experiments. Because if you're looking at cost of accommodation, traveling down expenses, and some little incentives, but want to move up and down, I think it would be good to do the tutorial. Let it be sharp and bright for we that are going to practice it, do it in our various homes.
Thank you very much. That's a brilliant suggestion. We would like to do a thing like that. Even those who have participated in this training, we are going to do this experiment, video it, and upload it for them to see how things work. And because bringing people down here from different parts of Nigeria will make a lot of cost and all of that. So let's make a promise that we'll do that. You see how we put the test tubes in different racks and how we undo them, how we prepare the nutrient broth. These are very interesting things. It makes my heart bleed that some microbiology students in Nigerian polytechnics and universities only hear these things by theory. They don't ever get to do it, or and it's a shameful thing. So it's a promise. We'll do something like that. Any other question?
No question, sir.
All right, so let's move to the next one, the chemical analysis. Yes, like I said, people boil their water, but that same water they used to cook beans. After they boil the water, the chemicals in that water are still there. So when they use it to cook beans, the beans get softened by absorbing all those chemicals into, and then we put oil and eat. Okay, so all the ions, heavy metals, and organic compounds that are in those water end up entering our body. Some of them cause cancer. Some of them make us have a kidney failure. Some of them make us have heart failure. And like I said, doctors are busy in the hospitals treating, trying to recover people of all those kind of challenges. Whereas if you have paid attention to the water we consume, we'll not be spending all those kind of monies. Those economy body will not be on us. So why don't we, from the water, start making our lives? Because you are healthy as much as the water you drink, and water is life. Okay, so what are the equipment we use to analyze for all these ions, metals, organics in our water? One of them is atomic absorption spectrophotometer. Another one is Fourier transform mass spectrophotometer. Another one is HPLC, gas chromatography, ion chromatography, mass. So these names are very big, but they are not fantastic. They are not things you cannot understand. And by God's grace, at Bioscird, we have a school of instrumentation and analysis. We like to make people understand what these instruments are, how they work, and they're very simple things to operate. Okay, so as for atomic absorption spectrophotometer, what that one does is it measures the amount of metals that are in your water: calcium, magnesium, lead, arsenic, all these metals that can dissolve in our water and we keep drinking them. This machine will tell you the quantity, what's the parts per million that you have in that water. And then when we compare it with acceptable values, we know whether that water is good enough or not. Okay, so the picture I've put here on the screen now just tells us what we have inside that machine. Okay, if we remove the outer casing of that machine, this is what you have inside. You can see where you put your sample, how the sample goes into that chamber, how the sample is heated up. Look at where the detector that will detect calcium, magnesium, and then it is transmitted to a computer. That computer there's a software in the computer that will pick all those stuff, and then it send it to a printer. The printer will bring out the chart that we can use to work and know the amount of all this. So they're very simple things. It's only our labs in our schools are empty. And these things look like there are things you see in Japan or in heaven. Okay, let's move on. Fourier transform infrared is just like this other one too. This one works on infrared absorbance. When infrared light bounces on some bonds, maybe carbon hydrogen or carbon oxygen bond, it causes vibration. There's a stretching and there's a bending. When that stretching and bending happens, we can quantify, we can measure the energies involved. Okay, and so